So today I'm going to talk about that how we can develop plant cell-based bioprocesses for production of such high-value phytomolecules. So I'll not get into the details. I think with the material which you have already received, there is plethora of information on the commercially viable phytomolecules on which plant cell-based bioprocess can be made and developed. And also what are the current molecules which are already in the market, which are derived from plants. And the wide range of industries in which they can be used. Now, I did hint upon what are the limitations of natural plant extraction, which is very clear. Here, I'll just reiterate because this is belongs to secondary metabolism. As you would have heard from Professor Nathli, the sometimes these products are produced only as per requirement. They are not always present in the plant. Sometimes they are produced because the nature would not do a compromise with its primary functions. So it is very obvious that only a small amount of carbon flux might go into the required secondary metabolism. And that too is sometimes temporal and spatially distributed. So therefore, in general, you would find the concentrations very low in nature for these phytomolecules. Concentration here, it means yields rather than concentration. Now, because of the limited supply, because it is geographical dependence is there, climatic dependence is there, so obviously there is limited supply. Many of these are rare species. Many of these are already endemic species, not easy to procure. So that becomes a limitation to extract out the phytomolecule. They need specific environmental conditions. I'll give you an example. We were working with a small class of plant peptides called as cyclotides, and we collected some of the, we were working on the medicinal plant called Viola odorata. Its common name is Banafsha. It is used in indigenous system of medicine in Unani especially for cough, cold, and also for sleep disorders. And there was an indication in literature that they have a rich repository of these cyclic plant peptides called as cyclotides. So, in fact, we were among the first ones to create a in-house protocol, FTMS-based protocol to identify these very low volume molecules from the Indian plant variety, which was Viola odorata. So we collected the plant biomass from Harwan, which we called as Harwan variety, which is in Srinagar. And then we collected some variety from Ooty. There was a horticulture there, herbarium where they were maintaining it. And then we brought some along with the soil, and we tried to see if they could survive in Chennai, in IIT Madras horticulture. So after 3 years of maintenance, and then we started trying to identify and decipher what kind of cyclotides are there in the biomass. They showed very, very different profile of cyclotides. The Srinagar variety had shown a different profile. There were many cyclotides which were, the maximum were present in those plants which were for 3 years being maintained at IIT Madras horticulture. In fact, the plant is not known to grow in this season. So this also gives an indication that the secondary metabolism is also a function of the flora, fauna, the temperature, the altitude, environmental conditions. So it's obvious that they become a function of their yields would be dependent on the conditions. So area under the plant, obviously, as Professor Nathalie also mentioned, if land procurement, there's always a competition between food crops and the medicinal plants for use. Long time growth. I gave you an example of Nothapodites pneumoniana. It does take 7 to 8 years to reach to maturity, where non-invasively it has to be taken out from the bark of the tree, camptothecin. But so there's a limitation and there's a time involved. So obviously the productivities then go low. For example, Azadirachta indica. The azadirachtin is extracted mainly from the seeds, but it's twice in a year. So one has to wait for that long and then harvest the seeds and extract out azadirachtin. Non-uniform. Now it must be very clear to you that why it is non-uniform and inconsistent. Chemical synthesis. from the very fact when you saw those complex structures and the complex biosynthetic pathways, she did mention that there are sometimes 30 to 40 steps of biosynthesis. So it becomes very complex, more than one chiral centers, very complex chemical synthesis. In fact, total chemical synthesis in research might have been published, like in case of camptothecin. But to the best of my knowledge, still commercially, total chemical synthesis is still not well known. And so it is because possibly of the low yields, maybe the racemic mixtures, the toxicity of the byproducts, the harsh environment, and of course, because of this, it is not a greener technology in sustainable development. So, what is the alternative? One of the alternatives is plant cell technology as well, wherein one can develop plant cell based bioprocesses which are scalable, which are amenable to optimization to produce these phytomolecules uh, in large amounts to fulfill the demand at any time in any part of the world. So the advantage is there can be control of supply because these are reactor driven controlled environment processes, high growth and productivity because these are amenable to optimization. There is a chance where you have better productivities because then you can have continuous production all throughout the year from these biomass. Reduction in time and space, obviously, because then you don't need a cultivable land. Strain improvement, cell line, what we were talking about as metabolic engineering, even plant cells are amenable to cellular engineering. So you can always manipulate the cell metabolism to enhance the flux towards the desired product. You would have heard from her talks that at any given time, how many different class of secondary metabolites a plant can produce. So the same carbon flux is diverted to so many different branches. So if you have to, and no wonder why the yields are low. So if you have to improve the yields so as to match the productivities, then you need to tweak the metabolism such that preferentially to your desired metabolite, the carbon flux flows. Ease in extraction, we did observe in case of cyclotides. When we had earlier, so we made a comparison of in the extraction, although this was relative abundance, what we found was in the plant biomass which we had collected from Harwan variety, which was in Srinagar, we found that two major interfering small molecules, linearyl and linearin monoacetate, were interfering with the the cyclotide pool. We were finding it very difficult to extract out preferentially only cyclotides. But when we developed the in vitro cultures, when we did the similar analysis, they were completely undetectable. And what we could get, it was only 4% of small molecule impurities vis-a-vis 96% of relative abundance of cyclotides in the extract. So that's also an opportunity when you work with in vitro systems that you may have ease in downstream processing. So what do we need to develop a plant cell based bioprocess? We need first the cell line or the callus line. So for which we need an explant. Explant is what? An explant is any plant part. Although I say any plant part because the plant cells are totipotent in nature. But when you are looking to develop a high yielding cell line, any guess which plant part will you be looking for to begin with? Which plant part you should be looking at? What earlier investigation should be done? Or you should just randomly pick up any plant part? What is the logic? Why would you say meristematic? We don't have to again it is already we can use it to make our plant. Okay, but the idea is to keep it at the cell level itself. We do not want to create a whole plant, isn't it? Yeah. It will show a good growth properties. Growth properties could be, yes, meristematic cells, they have better growth rates. But right now, what is the objective? Why are we looking at that's also a very good logic. So when we are looking at screening and selection of a cell line, both the factors are taken into account. One is the growth index and the product yield. So now what is done is you first do a quantitative analysis of various parts of that plant. First, you go back in literature and you try to find a cue. Which plant variety or a family is known to produce that class of molecules or that particular metabolite? Is there any literature available? That cue you pick up. You go and try to get hold of that plant variety. You now see and take cues from literature also and do a quantitative estimation where the product content seems to be maximum in nature. So many things, many cues you should pick up from nature. That's why in my lecture I was saying that why do we want to know that why is the plant producing it? Where is the plant producing it? How is the plant producing it? So as to you can adapt that in the lab for more targeted approach towards yield or productivity enhancement. So you go and you do a screening of which explant you'll use that as an explant, which in nature is supposedly giving you higher yields of the phytochemical. So explant is selected. Now what do you need? You isolate the cell line from there, assuming that the product yields are high. And also at the same time, the growth rates also have to match. So at any given time, when we'll come to how callus is induced, these are de-differentiated mass of cells. So which means that they'll have, because you'll use an explant to generate these callus lines, they can be heterogeneous in nature. So that's why a second step of screening would be required to check which among these callus lines is giving you highest yield and highest growth index, growth rates. So that becomes your choice of the cell line, chosen cell line to begin with. Then what do you need? Then you need to grow them, mass multiply them. So what do they need under heterotrophic conditions? Now they are no more in nature. So you will be needing to provide them medium components. So what should be done? You need to optimize the medium components so as to maximize the growth rates as well as your product yields. Then obviously being an in vitro system, these are done under aseptic environment, monoseptic. So this is the steps in short which are taken to develop a plant cell based bioprocess. How do we begin? As I said, we'll have the target plant, we'll take the explant, we'll generate the callus line. From this callus line, we do a selection of higher yielding callus cell lines. Now, then we go to cell suspension culture, and you can see a cell suspension culture which is somewhat mimicking microbial suspensions. Although ideally it has to be uniform, but there are certain limitations which have to be overcome, wherein again your media manipulation helps. It is easy to scale, it is easy to optimize any process conditions if your cell culture is as uniform as possible. You have smaller aggregates, there is less of aggregation. But by nature, the plant cells tend to aggregate, to stick together. So at this stage, what we do is we do yield improvement strategies, we do cell line selection. Yield improvement strategies will include your media optimization, culture conditions optimizations. Your yield improvement strategies will also be very, very targeted towards your phytomolecule, like your elicitation and precursor addition. I'll be coming to those strategies, which will be linked to your knowledge of the biosynthetic pathway and why and how in nature this molecule was produced. So therefore, all that knowledge of yours will be implemented to find the best precursor and the best elicitor which can lead to maximum yield enhancement in these cell lines. Now, elicitors, as I said earlier, these will be molecules. From where are you going to get the cues if someone remembers? From dead cells. No, the cues. How will you pick up the cues for what can be a good elicitor for this particular system?
From the wild, ma'am. We look at wild clues from where those stress responses are coming, and then we pick up those molecules as
So you use those stress inducers as your elicitors in laboratory conditions. So that is done at a shake flask level, at a smaller volume. Now, it is at this stage also where you generate data, generate lot of data. You can do transcriptome analysis. You will do all multi-omics here, which will give you a lot of information on that cell line and what in the absence and presence of an inducer. So suppose you are trying to decipher a biosynthetic pathway, like for example, what Professor Nathley was mentioning. So if suppose you want to identify the steps, some of the cues can be obtained from here. If something by adding an additive is inducing the secondary metabolite production, and if you do transcriptome analysis, a comparative differential transcriptome analysis, you should be able to hit upon which genes might be responsible for this elicitation to happen. So you can do all that data generation at this level. Then once you have optimized the process at the shake flask level, then you come to a lab scale reactor level, like what you do in microbial fermentations. The technology for fermentation remains the same. You'll come across in literature, people have coined the word called plant cell fermentation. Now, fermentation is no longer only yeast and anaerobic conditions. It is bioreactor cultivation. So this can also be termed as plant cell fermentation. Now, when you have the process ready at the shake flask level, what can be the limitations of the shake flask? Why is there a need to come to a reactor? I can have multiple shake flasks. Anybody? Absolutely. So you need to scale the process and you need to control the process at their optimum values. Not all parameters are easy to control at the shake flask level. So you bring it to a batch reactor level first. Now, the batch reactors, although it may seem very easy, and that is the mistake, what generally, why do you think until now, when the world is gearing up to plant cell fermentations and in various countries, I showed you a long list. People are doing plant cell fermentation, but somehow we are lagging behind in this. So one of my personal opinion is that mostly you will see the fermentation industry is geared towards microbial fermentation in our country. Now, there has to be a rational way of design. So what we are doing, we are trying to do a hit and trial, trying to adapt microbial fermenters for plant cell fermentation. Now, that is where the lacuna lies. If you'll try to adopt what is existing for microbes and imagine and assume that this will be very well replicated for plant cell cultivations, it would be a hit and trial and your luck. It may work, but most of the times it may not, because you have not understood the requirements of large scale cultivation of plant cells. One, how is plant cell cultivation different from microbial cultivation? And then come back to those bioreactor design parameters which are impacting the requirements for plant cell cultivations. So that is what we do at this bioreactor stage. We design the bioreactor for plant cell cultivation. And when I say designing the bioreactor, please make note, it does not mean the geometrical parameters only. It means the fermentation parameters as well, the feeding strategies as well, any operating parameters at the reactor stage which would lead to high productivities of your product. So that is what we do at the reactor stage. Now, everything, whatever we are doing initially is because of ease. We do it at batch level. But there are certain limitations of the batch fermentation, like those who are aware and those who are not aware, for their benefit. The batch fermentations have a limitation that it's a closed system. So you are left to give large amount of substrate if you want to have large amount of product. That's how the logic goes. But sometimes what may happen with that logic, the problem may be that if you get too much substrate to a cell, that may be detrimental for the cell. So where you will see that against your expectations, the product yields will not improve, but rather decrease because of the viability loss, because of the biochemical activity compromised. So once the upstream portion is done, we also at this stage try to work on the downstream part, which is your extraction of the phytochemical. Because all these parameters, when we are optimizing, are being geared towards your desired phytomolecule, the downstream processing is expected to become easier than what you would expect when you are taking a natural plant parts. Now, what do we do to enhance productivities? I told you that to bring in economical feasibility in any kind of bioprocesses, the productivity has to be improved such that your production cost is at least is able to match the product cost to begin with, and then you can use economies of scale to have more benefits. Now, what all we do to improve productivities? We can optimize the media composition, we optimize the environmental factors, especially some of the factors will be very targeted towards plant cells or tissues. Like in case of microbes, generally bacteria, let's say if you are doing bacterial fermentation, you don't bother about light. But in plant cells, you might have to look into the impact of light. Having said that, because these are in vitro systems, generally we try to make them heterotrophic. Now, it is important to also investigate that what impact is light going to have on the secondary metabolite synthesis as well, apart from the biomass. Now, for example, now again, you should take cues from nature. If a phytomolecule is being produced in the rhizosphere, in the root system, there is a good chance that you must try out total dark conditions and see what impact does it have on the phytochemical synthesis. Because this is linked, might be linked to the expression of the enzyme, biosynthetic enzymes. So light impact on the secondary metabolite biosynthesis should also be investigated to see if that can also improve your phytochemical yield in these cell lines. Addition of precursors, I'll come to the details. Precursors, just to let you know here briefly, these are your intermediates of the biosynthetic pathway. So what Professor Nathalie was telling you yesterday, if the logic is that if you keep adding the precursors, it would drive the rate of reaction forward. Now, if it is driven forward, there is a good chance that you should be observing higher product rate formation. So with that, precursor addition is done. But of course, there is a catch. I'll come to that. All these parameters additions, when we do, there are multiple things which have to be kept in mind and considered and optimized before you come up with an optimum strategy of these additives. Similarly, elicitors we add. All this is done to enhance productivities. Now, specifically talking about elicitors, because elicitors are mostly geared, will be stress inducers. Now, anything which is inducing stress and thereby inducing the secondary metabolism might be a compromise for the biomass. So, one has to carefully look into when and how you add these elicitors. So therefore, time of addition, the time of exposure with the cell line, all these things should be kept in mind and optimized if you really want this strategy to be successful. Till date, to the best of my knowledge, elicitation is one of the best strategies. You can go back and check in literature, which can lead to maximum secondary metabolite or phytochemical yield improvement in these cell lines, apart from cellular engineering. So they don't come under transform techniques. So this is a non-transformation technique by which you can get as high as 5 to 10-fold enhancement in the phytochemical yields. So it's a very good technique even now. Then immobilization. Those who have already done fermentation, maybe you have already read about microbial fermentations. Why do you think immobilization can be of use in plant cell fermentation? Any guess? For continuous fermentation? Okay. So what will be the benefit of it?
So in continuous fermentation, there is no need of inoculating multiple times. So it's a batch fermentation. It's not a batch fermentation.
So what is the ultimate benefit?
Oh.
What is the ultimate benefit? No, we are still at plant cell fermentation, so we are not. No, he is right. So continuous, I want to know the end result. Why are we doing? Even if the advantage is that we don't have to inoculate every time because it is continuous. So, but what is the advantage ultimately because of that? The productivities are higher, isn't it? For the product. So, why and in what scenario? Do you think that in every scenario continuous fermentations are good? Where you think, what are the prerequisites for this to work on plant cell fermentation? The product has to come out. It is a continuous, it's an immobilized system. You cannot be the product. First thing, the product has to be non-growth associated because the cells cannot grow. The prerequisites would be that and most of these secondary metabolism derived components, you will see an inverse relationship with the biomass. So that is a good chance that that will be met. What else? How else you think an immobilization can work with plant cell fermentations? I'll talk about the differences, but just for your information, I just said that plant cells tend to aggregate. And someone was just mentioning that as the plant would grow, this secondary metabolism is a specialized function. It's a specialized function. It is an expression of higher developments. You saw now with how evolution, mostly the secondary metabolism has diversified and has increased. So for survival. Now, in this case, if suppose the secondary metabolite is to be required, then the cell biomass cannot grow under the immobilized system. Now, the secondary metabolite has to be, therefore, extracellular. Most of these secondary molecules are intracellular. They are stored in. You lyse the product. So then what is the catch? How do you make use of this strategy? The cell-cell contact has been taken because these are higher order functions. So you also observe the more you give them a feeling of cell differentiation or cell-to-cell contact, you would see that the phytochemical yield may also improve. Now, if the cells are being immobilized, they are in the vicinity, that might help in the secondary metabolic synthesis, bolide synthesis. But the third criteria is that it has to come out for it to be collected in the spent medium. It's a continuous fermentation, like cell retention rather. So now you make use of another strategy which is called as cell permeabilization. That is where it will help you. And the other added advantage of cell permeability enhancers, these again, you will have to take care that in what concentrations they are added such that permanent damage is not done. The viability is not compromised to a large extent, but at the same time, they are reversibly made permeable. So if they are made reversibly permeable, the product will be able to come out of the cell into the spent media. Now, if the product is able to come out of the cell in the spent media, that would facilitate your cell immobilization systems or cell retention continuous cultivations, and therefore will be able to get higher productivities. High density cultivation, of course, because these are scalable systems, you select a suitable scale up criteria and then you can scale these systems in the reactors. Now, where does the role of mathematical modeling come in the bioprocess? Anybody? Those who have studied bioprocess in some way. The tools and techniques remain the same. Okay, wonderful. So which means in order to establish the kinetics, now if you want to describe the kinetics of your cellular system, you would be adapting some mathematical equation to fit your data into. And once you have fitted the data, which means you are assigning values to the mathematical equations constants. Once you have assigned numerical values to the coefficients in that mathematical equation, and it has given you a good fit, which is extrapolatable, then it is a model. Now, she was saying that it's important, it will be useful during growth rates, not only growth rates, but it can be useful to know substrate utilization rates. You also need to find growth rate is equivalent, doubling time. You want to know about how the cell is growing, how the cell is consuming the substrate per unit time, and how is the product getting produced per unit time. That is what is kinetics mainly. This is what we are concerned of. Now, why do you think it is needed? Why should I be concerned of this? Good, very good. So, if I can just explain it further, why do we need to know kinetics? And where do we need to know kinetics first? Is at the shake flask level itself. Why do you need to know? Because then you are understanding your system under these given conditions of culture environment, under the given set of environmental conditions, media conditions, additives. How is my growth rate of the cell? How is my product formation rate? How is my substrate utilization rate? Now, once you know this, you can have a profile. And once you make it into form of a model, what power do you get in hand? The power of prediction. Like giving you a small example. If we have y is equals to mx plus c, so it is a relationship between y and x. What power do I have now? If I'm able to get the value of m and c, I have a straight line equation. Now I know that this is a given straight line equation under the given set of data, which I used to get to this equation. Now, what power do I have? Even though that given set of data did not include all the data in the space, but now I have the power to keep changing x, which is called as independent parameter, and see the effect on y, which is my dependent parameter. Now, in fermentations, what are my dependent parameters? What am I interested in? Why am I calling them as dependent parameters? First thing, my product, my substrate, my biomass. Because substrate is linked with my production cost. Product is also linked with my production cost. Biomass is also linked with my product formation as well. That is why kinetics is important. It will help you to predict X, S, and P with respect to time under given set of conditions. So that is what we make use. What is the other advantage of mathematical modeling or this prediction? What is the advantage? Why am I saying that we get hold of we get the power of prediction? So, these are very, very big and ambiguous terms. I really wanted targeted answers. So, scaling happens a lot of things. So, why do you think prediction power is important? Just be very simple. Why do you think prediction is important? Why should I be bothered about having a predictive power? That I can do. Why prediction means without doing anything, I'm able to find what is the impact. Obviously, optimization, but optimization, an experimentalist can say, I'm anyways doing optimization. Why should I be bothered about the predictive power? Why? Hit and trial. Wonderful. So, why is this power necessary? Because this will avoid hit and trial. This will avoid a lot of energy, time, and resources spent in optimizing bioprocesses. And that is what industry is interested in. Do you think industry has time for, as a PhD project, to work for 5 years and wait for thesis to come out and see how the product is running? It is huge time to market. The incubation period in our ecosystem is 18 months on paper. Of course, for a biotechnology driven startup, we have come to an understanding that please have patience. There will be some time to market, but they cannot be waiting like a PhD thesis for a technology to be developed. So where do these things come in play? They will reduce the number of experiments which you would be required to do to reach to better productivities, enhanced production. Any model is a model only. Having said all this advantage, these are not, there is no perfect model. There's always scope for improvement. But the advantage of modeling is that you can use the model to enhance the production at the reactor stage, improve the productivities at the reactor stage. You can use models to improve the yields at the cell stage when you are doing cellular engineering. I'll give you some case studies later half of the course when Professor Nathalie will be talking about the strategies, how you can tweak the metabolism, what are the tools available. Coming to the first step, screening and selection. We generally use these two terms interchangeably, but technically speaking, they have different meaning. Plant cell cultures, they are heterogeneous because, of course, what happens that how do you create callus? You take an explant, you create a wound. So callus formation is a wound response in nature in these plants. So whenever a wound is created, the wound response sets in. The biochemical signal which sets in is it will try to revert back to its meristematic state. They will mass multiply, they will try to cover the wound, and then suberin wax, all that coverage is done. But what we do in lab is we do not allow it to stop. If we create that wound by cutting the explant edges and we give them, so what is responsible? Generally, these biochemical signals are coming from the combination of hormones. So we manipulate the medium conditions such that this can indefinitely keep multiplying. That is what you get as callus line. So what happens? Although this is easier that yes, you'll take an explant, you'll generate a callus line, and now you think you are ready to take that callus line. But because of when you create that cut on the explant, a number of cells are getting exposed to the medium. And not all cells are similar type. So all the callus which you will get as one bolus mass generally is a heterogeneous mass of cells. And therefore, as you will continue to subculture them, they may show some disturbance in the yields after continuous subculture. Moreover, on top of that, because you have used de-differentiation process and organized cell, because organ you had used, so these were well specialized cells. Specialized cells means these cells were not meristematic by nature. Because of the artificial medium environment, you have forced the cells to reprogram themselves, which we are calling as de-differentiation. So they are more prone to genetic instability when you keep subculturing them in the, which we call as soma-clonal variations. That's a kind of impact, the epigenetic stability, the impact of the environmental conditions on the cell nature, biochemical nature of the cell. So there is a good chance that even though you have had from different, let's say you collected 4 leaves from the same branch and you had created 4 different explants, you have made 1 centimeter square of explants, you put it on a medium, all these callus lines will not be of same nature. These are considered as different callus lines. Now, all these callus lines which you have in hand, then how do you do screening and selection to get to higher yielding callus lines? How are they different? Screening is a passive technique by which a number of cells are analyzed for the desired trait. Now, selection is something which is invasive. You are only going to allow the best performing cell to survive. The rest should automatically not able to grow. That's a invasive technique. Then screening, otherwise, on the contrary, is a technique where you are preferentially picking up the callus line, which is showing you better productivities. Now, where do you think this can be of use? One is that you first allow them to grow, then you do quantitative analysis of your product, and then you screen the callus line. Where do you think both the techniques can have different applications? Or if you have callus lines, you can apply all. Everything is taking time. Plant cells grow slow. So what cues will you take from nature? where your information about the phytomolecule will help you. Any guess? Where do you think screening will be very straightforward, which is a passive technique? How do you know? No, which is very easy. Something where you can do it quickly. So I'll prefer screening and I'll get to the highest yielding, possibly highest yielding cell line in hand. So there are many phytochemicals, as Professor Nathalie was mentioning, which have color to them, which are responsible for colors, different types of colors. So when you generate callus lines, they will also demonstrate color if they have that capability in them, isn't it? Like what you can see is in saffron callus lines. It will show you, not all callus lines may show you the same color. Some may be white, some may show you the orange color, some may show you deep orange color. So which one would you prefer, which will screen to take it forward? Because the secondary molecule is known to be responsible to give that color. So that is screening. Selection, where is it used? If suppose you are looking for, selection is an invasive process. So what will it be needing for you to do? Suppose you are looking for something which is resistant to, let's say, salt stress. I'm just giving an example. Any kind of stress. So you would like put them with that stress inducer in the medium and then see which one survives. That is possibly going to have the maximum resistance. This I've already told you. Now comes cell suspensions. So generally cell suspension can also be used for cell line selections because it will help you to further refine your heterogeneity because it's fine suspension. So it will, especially in case of selection when you are doing, only those cells will survive which will have that property. So then finally, when you take these cells and bring it to the solid medium to generate callus, it becomes more synchronous in nature, more homogeneous. This entire process may take anywhere between 4 to 12 weeks, depending on the species, the plant species with which you are working. Now, somebody was telling me we don't have to inoculate. Now, there is an importance in fermentation of inoculum, the nature of inoculum. Now, what is important is the inoculum density, the inoculum, which means inoculum size, the age of the inoculum, of what age stage are the cells. So what is an inoculum? Inoculum is the initial starter culture which you use to mass cultivate or further grow the biomass. Now, what has to be, how is it different from microbial cultures is that plant cell cultures, some minimum amount of inoculum is sometimes required. When you're especially working with organ cultures, we were working on hairy root cultures for azadirachtin production. What was observed was until unless you have the minimum number of roots given, size of roots given as inoculum, the growth doesn't begin. So there is that minimum inoculum density which will be required for growth to happen. So this is for your self-study. I've already told you about where screening and selection can be used, what the various examples in case of a colored callus or if you're looking for a resistant cell line. I'll read out this example where plant cell culture synthesize biotin from the precursors pimulic acid and alanine. High concentration of pimulic acid is said to be toxic to the cells while they can tolerate high levels of biotin. So biotin was the end product. Biotin is getting produced from the precursors pimulic acid and alanine. What strategy was used is high concentration of pimulic acid can be toxic to the cells. Now, one of the ways of cells, if they are having that capability to protect themselves would be to quickly take it and metabolize it into the end product. So that gives you a logic that if you will add that as a stress inducer, and if the cells which are able to survive, possibly they will be higher biotin producing cell lines. Now light is an important factor, because there what you can take care of is the photo period, which should be optimized, and also sometimes the wavelength of light. You'll come across a lot of literature where secondary metabolite synthesis might be a function of a particular wavelength of light. It can get enhanced. Even in agriculture, this is a strategy to improve the production. So it is a fair idea to also look if a particular wavelength is enhancing your product yield multiple folds. But it is highly species specific thing. So for every species, when you begin with, this parameter should be investigated. And of course, having said that, light, you will be then, if suppose you are working on 16 to 8 hours light dark, you have to consider the cost component, because then these will be photobioreactors. The scalability will have to be looked into. So generally speaking, it is not a bad idea to see if you can convert them into completely heterotrophic cultures, so that you can avoid light, and especially the photobioreactors. And sometimes you may also observe that under complete dark conditions, it may become a stress the cells, which can also lead to enhanced secondary metabolite production. So therefore, to investigate the effect of light is an important parameter. Then talking about, obviously in fermentations, all the fermentation parameters are important. Mixing is an important parameter. Homogeneity is important. So therefore, mixing is important. Now, in plant cells, in comparison to microbes, they are relatively more shear sensitive. So microbes, you will find fermentations run at RPMs which are greater than 400 at reactor scale, 400, 500. But you cannot afford that high speeds for plant cells, because it has been observed, if you'll see literature, they somewhere range around between 80 to 125 maximum. Now, the more you go beyond, you keep increasing the RPM, the cell viability is compromised. So one has to take care that the cell viability should not be compromised to an extent that the biomass is impacted, and therefore your biochemical activity. So there has to be a fine balance between the mass transfer requirements and the cell viability, the shear effects. And because they are relatively more sensitive, unlike microbial fermentations, it seems to be a crucial factor to be kept in mind. Of course, the volatile impact can be there. If you have very high RPMs to fulfill the mass transfer requirements, sometimes your gaseous volatile components, which may have an impact on secondary metabolism, like CO2 or ethylene, let's say, which is a gaseous hormone, which may then escape from the headspace, might compromise your secondary metabolite synthesis. pH and all, these are well-known fermentation and culture parameters which you must take care. So I'll not get into the details of it.